Hello everyone, this is a video for Unit 2, Cells Development, Biodiversity, and Conservation. This is a revision video, and this is going to be part 1. Okay, so in this video, which is part 1, we're going to be focusing on topics from 3.1 all the way to 3.5. So that's knowing that all organisms are made of cells sharing some common features, and all the way to talking about the differences between eukaryotic cells and prokaryotic cells, and of course the RER, the Golgi apparatus, and other organelles present in the cells. And this is also part of the Unit 2, which is also topic 4, plant structure, function, biodiversity, and conservation. This is going to be covered in later videos. Okay, so just a quick recap on cell structure, reproduction, and development. So first of all, we have organelles. Organelles are compartments within a cell that carry out a task. And then we have cells. So cells are the basic functional and structural unit in living organisms. Then we have tissue. Tissue is a group of cells that work together to perform a particular function, okay, so like muscle tissue. And then we have organ. So an organ is a group of tissues working together to perform a particular function. Your liver is an organ, okay, it breaks down harmful toxins, okay, that's its function. We have the organ system, so your digestive system, which is a group of organs working together to perform a particular function. Yes, these definitions are required for you to know for your A-levels. Even though they're all GCSE knowledge, they're also required for your A-levels. Okay, so organelle structure for eukaryotes. So eukaryotic cells have a more complex ultrastructure than prokaryotic cells. Ultrastructure just simply means the internal structure of cells. You do not need to know the meaning of ultrastructure for your exam. So the cytoplasm of eukaryotic cells is divided up into membrane-bound compartments called organelles. Eukaryotic cells in both animal and plant cells are, number 1, they are membrane-bound organelles, okay, including the nucleus. And they also have larger ribosomes than prokaryotic cells. The ribosomes for the eukaryotic cells are called ATS ribosomes. So the key difference is that animals contain centrioles and have microvilli, okay, and plants have cellulose cell walls and large permanent vacuoles and chloroplasts. So animal cells and plant cells are both eukaryotic cells. However, animal cells contain centrioles and microvilli, whereas plant cells contain cellulose and vacuoles, and of course chloroplasts, and animals don't have that. Okay, now here's a picture of an animal cell. You can see we have the microvilli, the Golgi apparatus. We have microtubules, which are radiating from centrosomes. We also have ribosomes. In animal cells, they're going to be ATS ribosomes. So what's good practice for you to do is to draw this out and simply label each part. And when you label each part, you can, for example, draw the Golgi apparatus or the Golgi body and just write what's the function of the Golgi body in a bullet point. You can just say modification of polypeptides. So I can just draw over here with my line, and then I can just draw a star and then just write modification. So drawing is one of the most helpful ways that helped me study different parts of the cell and memorize different parts of the cell. You can also do the same thing with the mitochondrion, the rough endoplasmic reticulum, and the smooth endoplasmic reticulum. Okay, then we have a plant cell. So over here we have the tonoplast over here. It can also be drawn out in your exam through a root hair cell. So if you have a root hair cell that looks like this, the tonoplast is going to be a large part in the middle that looks like this. Because also a root hair cell is technically a plant cell, so they will and can ask you about it. So the plant cell is going to have plasmodesmata. So the plasmodesmata is going to allow cell-to-cell connection. I'm going to further explain this in the coming slides. We have also a Golgi body in a plant cell. We have chloroplast, we have grana in the chloroplast. Remember, the chloroplast is a double membrane, same thing as a mitochondrium. We have the nucleus, the nucleolus, and other parts of the cell membrane. Also draw this out and label it, and also make a star with the functions, which are going to be in the coming slides. Okay, so let's talk about the organelle functions. So we have the cell surface membrane. So remember that the cell surface membrane is made up of a phospholipid bilayer. And we have our tails and our protein head. Here you go. Okay, so the cell surface membrane, number one, it controls the exchange of materials between the internal cell environment and the external environment. And the cell surface membrane is also partially permeable, so meaning that some substances can pass through the membrane while others cannot. The cell membrane is formed from phospholipids. So it's a phospholipid bilayer spanning a diameter of around 10 micrometers, and it's also referred to as the plasma membrane. Now, the nucleus is present in all eukaryotic cells. The nucleus is large and separated from the cytoplasm by a double membrane called the nuclear envelope, and it also has pores. The nuclear pores, number one, they allow mRNA and ribosomes to travel out of the nucleus. Number two, they allow enzymes such as DNA polymerase and signaling molecules to come in. So there also contains chromatin. Over here in the diagram below, you can see how chromatin looks like. It looks almost like spider webs. So chromatin is the material which chromosomes are made out of. And chromosomes are made of sections of linear DNA that are tightly wound up around histone proteins. Now, the darkly stained region of the nucleus can be observed under a microscope. These regions are individually termed nucleolus and are the sites of ribosome productions. We have also the rough endoplasmic reticulum is connected to the nucleus and the nucleolus. That's because when it makes ribosomes, it's going to travel all the way out through the nuclear pores and it's going to attach onto the rough endoplasmic reticulum. Okay, now let's talk about the mitochondria. The mitochondria is the site of aerobic respiration in eukaryotic cell, and it is visible with a light microscope. If you use an electron microscope, you will see the outside of it. And it's surrounded by a double membrane, with the inner membrane folded to form cristae. Now, cristae is it's just foldings. Over here, you can see that cristae is foldings. So the matrix of the mitochondria contain enzymes needed for aerobic respiration, producing ATP. Okay, so aerobic respiration produces ATP. Now there are small circular pieces of DNA known as mitochondrial DNA, and ribosomes are also found in the matrix. These are needed for the replication of mitochondria before cell division. Now let's talk about ribosomes. Ribosomes are found everywhere. Remember, we have 80S ribosomes and 70S ribosomes, which are the smaller ribosomes in prokaryotic cells. Ribosomes are free organelles in the cytoplasm, or they are bound onto the rough endoplasmic reticulum. Ribosomes do not have a membrane surrounding them. Okay. Complex of ribosomal RNA are RNA and proteins. We have 70S in prokaryotic cells, 80S in eukaryotic cells. Now, ribosomes are the site of translation. Okay, now let's talk about the endoplasmic reticulums. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum is formed from folds of membrane continuous within the nuclear envelope. The surface is covered in ribosomes. So these ribosomes are bound on the surface. The role of the RER is to process proteins and fold the proteins which are made on the ribosomes. The smooth endoplasmic reticulum is formed from folds of membrane, but its functions is being involved in the production, processing, and storage of lipids, carbohydrates, and steroids. and it's smooth, so it does not have any ribosomes. Okay, now let's talk about the Golgi body. So the Golgi body also consists of flattened sacs of membrane, and it's distinguished by its regular stacked appearance, so it almost looks like a Wi-Fi signal. The role of it is to modify proteins and lipids before packaging them into Golgi vessels. So if you are writing out notes, the most important part to write out is this part. This part isn't really that valuable. Now, Golgi vesicles, number one, they transport the proteins and lipids so that the proteins can go through the Golgi body, okay? Number one, they're exported from the cell. So things like hormones such as insulins, okay? And they're put into lysosomes such as hydrolytic enzymes. So we have proteins that enter the Golgi body through the cis phase. They're going to be going through a series of modifications, and then they're going to be packaged. So over here in the trans phase, they're going to be packaged into vesicles. Now these vesicles, they can either contain hormones, they can contain enzymes. It depends on what the rough endoplasmic reticulum has produced. And then these vesicles are going to be transported to the specific location that they require, and they're going to bind onto the cell's plasma membrane, which then allows them to secrete whatever protein substance is produced. Okay, lysosomes. So lysosomes are specialist forms of vesicles which contain hydrolytic enzymes. So these hydrolytic enzymes, they carry out a process called hydrolysis, which is breaking apart substances and molecules. A vesicle is a tiny membrane-bound sacs that transport substances around inside or a cell, or for cell to cell. Also, they're involved in the process of exocytosis. So the vesicles are released by a process of exocytosis, okay? The role of these vesicles and lysosomes is to break down waste materials such as worn out organelles. Lysosomes are used extensively by cells of the immune system and in programmed cell death known as apoptosis, okay? Over here, we have a drawing of a lysosome. Okay, now centrioles. Centrioles are made of hollow fibers called microtubules. Microtubules are filaments of proteins that can be used to move substances around inside a cell, also as well as to support the shape of a cell. Okay, we have two centrioles at 90 degrees to each other to form a central zone, which organizes the spindle fibers during cell division. Centrioles are not found in plants and fungi. They are only found in animal cells. Okay, now let's talk about formation of extracellular enzymes. So organelles are membrane bound. They're involved in protein synthesis. So the organelles involved in protein synthesis are number one, the nucleus, where transcription occurs, ribosomes, so that's free ribosomes and those on the RER, which produce proteins in the process of translation. We have the rough endoplasmic reticulum, the Golgi body, and of course the cell surface membrane, where proteins formed within the cell are secreted. Okay, so we have the RER and the Golgi are involved in producing, packaging, and transporting proteins in a cell. This process can be used to produce and export extracellular enzymes. Okay, so over here in number one, this diagram over here. Now, number one is going to correlate with the step number one, and same thing with number 2, 3, 4, and 5, 6. So number one, the nucleolus is going to manufacture ribosomes for protein synthesis in the RER. Then what happens next is that the nucleolus also manufactures mRNA, which is needed by ribosomes to make proteins, okay? The ribosomes on the RER make proteins. The RER is going to process the proteins, which are then sent in vesicles to the Golgi body. The Golgi body further processes the proteins and sends them in vesicles to the plasma membrane. The vesicles fuse with the plasma membrane to secrete the finished protein product. Okay, now let's talk about prokaryotic cells. So bacteria are prokaryotic cells. Now prokaryotic cells are much smaller than eukaryotic cells. They are also different than eukaryotic cells by having or lacking, number one, the cytoplasm lacks membrane-bound organelles, so they do not have membrane-bound organelles. Number two, the ribosomes are smaller. They are 70S ribosomes. And also, prokaryotic cells do not have a nucleus, okay? They have a single circular bacteria, chromosome, and it's not associated with proteins because their chromosomes do not have histone proteins, okay? Now, a cell wall that contains glycoproteins, okay? Murin is also known as peptidoglycan. They have plasmids, capsules, pili, and a cell membrane that contains fold known as mesosomes. Now, plasmids are loops of DNA that contain genes. So plasmids contain sometimes antibiotic resistance. Now, plasmids aren't needed for the survival of bacteria. However, they do provide some advantages, such as being antibiotic resistant. Now we also have a capsule. The capsule is the outer layer, which helps protect bacteria from drying out and from attacks by the cells of the immune system. We have the flagellum, which enables movement. We have pilus, which enables attachment to other cells, and we have mesosomes, which are used in aerobic respiration, cell wall formation, and DNA replication. We have circular DNA, which carries genetic material found in the nucleotide. We also have ribosomes, which is the site of protein synthesis. And over here is a drawing that shows you a prokaryotic cell or bacteria. And here we have on the left side, the flagellum is sometimes present, the capsule is sometimes present. We have the foldings, the plasmid, and the pili. And the features that are always present are the cell wall, which is made of peptidoglycan, the cell surface membrane, the cytoplasm, circular DNA, and 70S ribosomes. And here's a table that's really important for you to know and understand, so make sure to copy this down and write it out. So we have the differences. Here's a summary of the differences of prokaryotics and eukaryotic cells. So generally speaking, the prokaryotic cells are smaller, eukaryotic cells are larger. Okay, now the genome, prokaryotic cells, their DNA is circular with no proteins. So prokaryotic cells, their chromosomes are not going to have any histone proteins, okay? Whereas eukaryotic cells, DNA is associated with histones, which are formed into chromosomes, okay? Now, cell division for prokaryotic cells, it occurs by binary fission, so it has no spindle fibers. For eukaryotes, it occurs by mitosis or meiosis and involves spindle fibers to separate chromosomes. Ribosomes are smaller in prokaryotes, so they're 70S. Eukaryotics are 80S. Now, organelles. There are going to be very few and no membrane-bound organelles in prokaryotes, whereas in eukaryotes, there's a lot of membrane-bound organelles. The cell wall of prokaryotes is going to be made out of peptidoglycan, and in eukaryotes, it's going to be either made of cellulose or chitin. Thank you so much for listening, and please subscribe for part 2.